My name is Thomas Langmann. I'm from Cologne in Germany. I'm heading the Laboratory for Experimental Immunology of the Eye. So that's a basic laboratory that tries to develop mechanisms and to understand mechanisms of the immune system in the eye with a focus on the retina and also related to macular degeneration and rare genetic diseases like retinitis pigmentosa. Actually, I was trained as a biologist and biochemist. Then went into laboratory medicine, and the next step was to go into human genetics. And with human genetics, actually, I was exposed to this vast group of genetic diseases of the eye and of the retina, especially. And the focus shifted from the more monogenic forms to the macular degeneration and also to diabetic retinopathy, recognizing that the immune system is a very important component in all of these different diseases. Actually, here we had a special interest group meeting focusing on the topic of retinal microglia, that is the resident immune cells of the retina. And this question we asked was, what was their function? Because actually in the last years there was a lot of research done how the microglia cells react to damage, for example to photoreceptor degeneration, in experimental models, also in AMD with different conditions of damage, for example light. And the special focus of this meeting was to tackle the different functions of these immune cells, whether they are good, whether they are bad. And we wanted to compose a panel of different scientists that have different topics in that area. For example, focusing on the migration of these immune cells, on the other side focusing on the things these cells secrete and produce that might harm the outer retina. And another aspect was, for example, the interaction of microglia with neurons, like photoreceptors. So we compiled different experts and tried to address the question, what is the most important thing, most important mechanisms that drives to the activation of these immune cells. And what was actually concluded and recognized was that there are many signals of dying or of stressed cells, photoreceptors for example, that then trigger the activation of these immune cells, and we were working out several mechanisms, including mechanisms how microglia can respond to dying cells, how they take them up, and whether they do good or bad, for example. That's a very good question, and actually I think that the immune system in general, not only in the retinal field or in the eye field, is a very attractive target for therapy. As we see from so many different applications, oncology, cardiovascular disease, modifying the immune system seems to be very elegant and a very good way of indirect modulation of a disease process, for example, of the retinal degeneration. And we think that the couple of next years we'll see some clinical trials with immunomodulation components. Some have already started, like for example minocycline here in the US, and some others are waiting to come where the different mechanisms are still to be understood. But in general, immunomodulation will be a very broad and very hot topic in the different disease areas of the retinal degeneration field. And I think that either alone, as immunomodulation alone, or in combination, for example, with anti-VEGF therapies, might hold some promise to delay disease processes. I think it will be drugs. It's now at the border of finding the targets, of identifying certain components, for example, immune cell receptors that can be targeted, for example, with antibodies. I mean, we know from the anti-VEGF therapy that antibody injection in the eye is something that is feasible. But also if you think about long-term processes, for example, in dry AMD, that takes years to develop and then also then in the end goes very fast, it might also be important to develop pharmacological compounds that are working not by direct injections, but maybe by just by taking tablets. And this might ask for some other very how could I say that, some very smart components that can be taken daily, and then they should prevent the activation of the immune cells over several weeks or years, but they should not harm the immune system. So they should not block immune responses in the eye or in the body somewhere else. And this is a big challenge, to find immunomodulation therapies that act via long-term, especially, for example, in AMD. Actually, there have been a lot of epidemiological studies that diet and compounds that affect the immune system might modify disease progression. One example is fatty acids. I mean, from ARED studies and from others, there haven't been very conclusive data that, for example, omega-3, omega-6 fatty acids really are beneficial. But what we see from a lot of animal models is that these fatty acids have a very strong immunomodulation part. So it might be that in the end, modification of diet, eating certain fatty acids might be beneficial. Also, other components like certain vitamins and some other components that keep your immune system healthy might be promising. On the other side, things that might activate the immune system over long term in a chronic fashion, for example, we all know that smoking is not good, but especially the oxygen radicals and the toxic microenvironment in the eye is now recognized as something that is very immunogenic. So actually, soluble immune components like the complement factors might even recognize cell surfaces that are damaged by oxygen radicals, by reactive oxygen species. So behavior that keeps you healthy also seems to keep your immune system healthy. Actually, there are, in the US, you can simply look it up in clinicaltrials.gov. There are some trials, as I mentioned, minocycline therapy is used for diabetic retinopathy and also for some monogenic forms of degeneration. But many of these immunomodulating therapies are just at the stage of moving into clinical trials. There are trials with complement factors, a lot of them, but this is something I would say that is a little bit different from the immunomodulation. The complement inhibition studies are meant to take away certain toxic complement components, for example, by using antibodies, by injecting antibodies. But this doesn't directly mean that you modify immune cells per se. So modifying the cells is something, what I explained before, that might need novel targets, for example, receptors on the cells or some interactions with photoreceptors that might be then modulated. And there we have some components that are now moving into clinical trials. One of that is, for example, as I said, minocycline therapy. Another one is polysialic acid our group is working on. So that's actually a sugar component that is known to be immunomodulating the cells without killing the immune cells, because this is something we don't want. We don't want immune cells to be dead or not functioning anymore. We just want to bring them that they do not something that is neurotoxic.